So moving right along, Kevin Hagedorn with Life Magnetics. Oh, good.
Hi, everybody. I'm Kevin Hagedorn, Dr. Kevin Hagedorn, and I'm here to talk to you about our company, which makes products for RNA sample preparation. So that means I have to first explain what the hell is RNA sample preparation. Sample preparation in biology is basically the process by which you make a raw biological sample ready for analysis. So you're a laboratory technician and you get a blood sample. The instrument won't just take the blood sample. You usually have a particular analyte you're looking for, which is probably in the cells, right? So you have to like blow up the cells and get it out of the cells, and that's sample preparation. It's the first and most important step in a lot of biological processes. It's the first thing. It's basically as common as test tubes if you're a biologist or doing anything with biological analysis. RNA sample preparation in particular is both a huge opportunity and a major challenge. It's currently 40% in molecular diagnostics, but an Endpoint News survey of the 500 biopharma executives identified RNA tech, gene editing, and gene therapy as the biggest growth areas. 3M's Partners, an Ann Arbor-based consulting firm, identified direct-to-consumer RNA testing is the highest diagnostic growth area of the next 10 years. So why is that? So you're all probably familiar with 23andMe, which is direct-to-consumer DNA testing, right? You send in a sample of your saliva, and they will give you predictions as to what sort of diseases you might get later in life. The issue really with DNA testing is that you're never going to get a different answer. If you do a DNA test when you're an infant, when you're a teen, when you're an adult, you get the same answer. RNA is basically the running programs. It's like DNA is a hard drive to RNA is the RAM. It's whatever programs are currently running. So if you have rheumatoid arthritis, for example, everybody has the genes for inflammation. But if you have rheumatoid arthritis, then your genes associated with inflammation are being overexpressed relative to the general population. So if I'm a drug company, I can give you a rheumatoid arthritis drug and see how your expression profile, first diagnose that you have the condition with an RNA expression profile, give you the drug, see how it changed after you started taking the medication, and then adjust your dosage accordingly. It's basically a DNA test, but it changes relative to your biological state, so it gives you better diagnostic information. The challenge is that the half-life of DNA is 531 years. The half-life of RNA is 28 minutes. So you have about 9 minutes to get the sample to the lab, which means there's no RNA testing, direct-to-consumer testing right now. We fixed that. How the product works, I don't have time for. It's a bunch of quantum mechanics. My business development coaches tell me I can't talk about that, which is unfortunate because it's frankly more interesting than this. But this is the product that we make. It's a particle which sequesters RNA. It's difficult to see here, but this is the stuff mixed into solution. If there's RNA present in the sample, it'll form aggregates, and the RNA is sequestered in the aggregates, which prevents them from degrading. We've compared sample preparation is very common. Almost every Fortune 500 biotech company has a product. Ours works better than theirs. This was a study funded by the Michigan Economic Development Corporation. We showed that compared to, here's part of our data, we can give you more, but we had better, in addition to it being able to sequester and stabilize RNA for 25 days, we got better yield and purity than competing products by Fortune 500 companies. Our team is myself. My PhD is in material science from the University of Michigan. I did semiconductor device physics. That's how I'm picking at these guys' EM thing. My co-founder is a molecular biologist, though. Andy Retter has done $2 billion in deals between life science tools companies and major corporations. His last two were Armune in November. Bob is CEO of Dexterity Diagnostics. He developed the first DNA testing platform. We have about $1 million in funding, most of which is from the National Science Foundation. And our ask is, we've got, I think 42, actually less than this now, ask Ken, remaining in our open 250,000 round. We're looking for sales reps. Our product retails at University Labs for, I'm going to go over, you don't need to tell me. Our product, yeah. So, I'm just not going to worry about it. Right. So, competing kits, the 250 reaction kits will go for about 1,200, 1,300. Yeah, just reset it. Our cost to manufacture 250 reaction kit is $35. So we have a large margin. We're looking for sales associates to help us sell to university labs, and teaching labs are excellent targets. So if you're interested in working with us as a sales consultant, we need that. And we've got, I think, 32,000 left in our open round. Again, ask Ken. And we're hoping to, because this is carried by a lot of biopharma companies, we're hoping to reach 500K in sales and then be picked up as a distributor by one of them. So that's our goal for this year. We're just going to stop there. That's it. Thank you very much. Questions? Right? 4 minutes for questions about RNA sample preparation. There you go. No. In fact, we'd prefer if you weren't, because the Detroit area and Ann Arbor, we've got covered pretty well. That's like where we're based, so please don't be there. Yeah. Oh, yeah, we're stable for 25 days. So normally it's a 28-minute half-life, but you want to, so there's this measure of the integrity of the RNA called the RIN value, and 28-minute half-life means half the sample is degraded in 28 minutes. So to get a RIN above 9, you'd ideally want to get it to the lab within 9 or 10 minutes, but we maintain a RIN above 9 for 25 days, so. We're not. So if when you're talking about, for example, when you're talking about a hospital, hospitals will just purchase whatever, like for example, University of Michigan Blood Pathology Lab, we interviewed Don Gachero, he's the director there. They basically have this big instrument from GE Healthcare that does all their analysis for them. And stuff like this is just a reagent that they refill. So the technicians see that like the light is flashing, and then they just reorder their sample from GE Healthcare. They're not really making a purchasing decision associated with this type of product. So our route to selling to hospitals is not going through the hospital, it's going through GE Healthcare or a distributor like them. And frankly, they already have products that do sample preparation, so the challenge is then convincing them that we're a big enough threat that they want to integrate our product into their product lines, which we're doing by targeting the university labs. Oh, yeah, so those aggregates, you pull these out with a magnet. So after you mix this in and it does that, you just put it next to a magnet and then you can wash out all the other biological material.
Anybody else? Yeah.
Does this technology have any application beyond RNA sample stabilization?
Fundamentally, our technology, so I'm a material scientist, semiconductor device surfaces. I got the idea from Bob, who was at the time CEO of Motorola Life Sciences. And our fundamental materials processing is controlling the carbon properties, the sp2, sp3 ratio on the surface and surface charge. I haven't I've got a couple of like ideas, but none are as big or as this. So
Is that a yes or a no?
that is a maybe. 20 seconds, one question.
So if you're looking for kind of sales associates in this area, is there a background you're looking for?
Biology would be helpful. Yeah, so you don't get too many of those, right? Like everybody just wants the Internet of Things. So if there's any biologists, come talk to me.
Is this accepted by physicians as a normal practice?
They don't, so a physician, if they're doing a diagnostic test, probably doesn't even see that this happens. This would be something that they would order a test, and then the sample preparation happens in like the laboratory diagnostic testing lab, which is like then not a, the physician is not involved in that like ordering process. This would be like, for example, if I create a diagnostic test and I'm doing like an LDT, I tell doctors to use my test, and then my test is certified in my lab, which is somewhere else, and then I use this as part of that process. So physicians don't really make a purchasing decision in terms of using a product like this.
So short, is RNA testing with actual patients common right now?
Yeah, because almost all viruses are RNA, so if you want to do an HIV test, for example, you have to do an RNA test, because all viruses are RNA.
Thank you so much. Oh, yes.